This is Proteins Take 2 because I had stopped sharing my screen apparently during take 1, and so it's just me talking without you getting to see anything. So we're going to try this again. Proteins are essential for the human body, but we have to ingest all of the amino acids that we need to make our proteins. You have 20 essential amino acids, and they all have to come from our diet. Thankfully, human beings have a varied diet. In fact, speaking of animal species and their diets, human beings are probably the most diverse in what they are willing to eat. We eat everything from fruits and nuts and berries to legumes to nearly any protein source that we could find on this planet so far is going down to eating snails and little barnacles, because we're kind of willing to eat almost anything as a species. Historically speaking, that hasn't worked out for some people because we found out that some foods on this planet are highly poisonous and toxic to us because somebody had to find that out for us because they were willing to eat anything. Proteins build. That is what proteins do. We are made of protein. You're 86% protein by dry weight. And we get it from our diet. So let me share this screen with you. Look, sharing okay. Proteins. Meat. Most people eat meat. Some don't. We have vegetarians, we have vegans, we have people who believe only in the Paleolithic diet, which is only things that are naturally found and in no way processed, so it's all raw. We have raw food diets as well. And a lot of times these typically do not involve eating any kind of meat. But meat is where we get a lot of our proteins from. Proteins are meat. Muscle is protein. So, you know, beef steaks and chicken tenders and lobsters and I hope you're not eating cow in raw form. If you go around cannibalizing cows, which you aren't a cow, so that wouldn't be cannibalizing, but if you are eating raw cow, I think you might need to see a dietary nutritionist. Milks and yogurts and cheese and sour cream and cottage cheese and all that. And then eggs as well. Protein, 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 protein. Proteins. You have proteins that do everything in the human body. Proteins start out as a single linear chain of amino acids. But as they grow and grow, coming out of the ribosome, they start folding on each other, on itself, and building secondary and tertiary structures that end up looking like this individual light brown, or this individual pink, or this individual light purple or orange. And so that's a single polypeptide. But there are many proteins, there are very few individual polypeptides that function on their own. A majority of our proteins have to work with other polypeptides to be functional, and this is called polypeptides. They have to work with other polymers of proteins to be functional, and these are called polypeptides. It's our quaternary structure. So we have primary, secondary, tertiary, and quaternary structures to proteins. We'll talk about that in this PowerPoint. Here you have Rubisco. It's a growth protein in some way or function. Okay, I don't really know what Rubisco does. I'm sorry. We have hemoglobin over here. It's 1, 2, 3, 4 subunits of polypeptides that are working together. And you can see here in the center of each hemoglobin unit is iron to carry oxygen. We have growth hormones. We have other types of proteins. The different colors up here in Rubisco tells us that this orange is an individual protein and this red is an individual protein, light blues, purples, and you can see all the individual proteins. This is actually a very, very large structure because of all the different proteins. Proteins work together to do what? Everything. Oh my goodness, proteins do everything. They're enzymes. we have many enzymes in our human body. Enzymes are the reason that we can live because they increase the rate of metabolic reactions in our cells so that they can meet our life processes on demand. If we didn't have enzymes, things would have to happen at a much more natural pace. We would die waiting for things to happen. We have enzymes like pepsin in the stomach, like DNA polymerase in the nucleus of the cells. Pepsin, in fact, breaks down proteins that you've eaten, and DNA polymerase makes new DNA. Proteins are involved in structure. The keratin in your nails and your hair, the collagen under all the layers of your skin. They're carriers and transport. Carriers such as hemoglobin in your red blood cells that carry the oxygen around the body as it is bound to that iron molecule in the middle of it. Aquaporins are for water transport through cell membranes. They function in cell communication by signaling each other with insulin and other hormones, and as receptors on the surface of cells for these signals. Proteins function in defense as antibodies floating around your bloodstream, waiting to come in contact with a foreign antigen. When the antibodies do contact a foreign antigen, they coat the antigen. This is called opsonization. And when opsonization occurs, it alerts these closest macrophages to come and ingest this antigen and break it down and then present those proteins on the surface of the macrophage cell. Movement. Actin and myosin are the two muscle subunits. So every single one of your muscles, if you get down to the very, very, very microscopic level, is actin and myosin working together to help contract and relax that muscle. And then as storage in seeds, like bean seeds, the proteins to make that bean plant grow. As you can see, proteins are the most structurally and functionally diverse group of macromolecules in the human body, and they are involved in almost everything. The structure of a protein, it is built from amino acids. You have 20 essential amino acids that you have to get from your diet. I don't know why I keep saying dietary. I did it last on take 1 too. These amino acids are bound together by what else but a dehydration synthesis. So we'll talk about the structure of amino acid on the next slide, but I do want you to note, every single amino acid is built exactly the same. You have an amine group, you have a central carbon with a hydrogen coming off of it, and then you have a carboxyl group, which is that carbonyl and hydroxyl together, and then you have an amine group, a central carbon with a hydrogen, and a carboxyl group. Amine group, carbon, hydrogen, carboxyl group, over and over and over again. That's an amino acid. The only thing that changes is this variable R group coming off this central carbon. This can be something as simple as a hydrogen or as complex as the ring structure. And there's 20 different versions. When you bind these amino acids together and form a long chain, you get a polypeptide. As I've already mentioned, the chain doesn't stay in just a linear form. The chain actually starts to fold on itself, first in like a 2D kind of structure and then into a 3D structure, so secondary and tertiary. And then a protein itself can be one or more polypeptides folded together and bonded together. If you have 2 or more polypeptides working together, then you have the quaternary structure of proteins. And this is growth hormones, and here's hemoglobin. And as you can see, these are large and complex molecules with a complex 3D shape. If you are trying to form a Rubisco and something malfunctions, you get a mutation in one of the proteins, you're not going to have the unique 3D shape that is necessary for that protein to function, and that protein will not function. The 3D shape has specific functions. You have these dips and crevices for reasons, and so it has to look that way. Okay, amino acids. Structure, once again, is that central carbon with a hydrogen. carbon has to form 4 bonds. One of those other bonds goes to that amine group. It's always amine group on the left, central carbon, carboxylic group on the right. Another name for carboxylic is, or carboxyl group is carboxylic acid. So amine acid, amino acid, right? It's an amino acid. It's a mean old acid. I know I've already used that joke, but hey. Then you have the variable side chain, the R group, which can be one of 20 different things hanging off that central carbon. Just as hormones and steroids function differently depending on the side groups that we have coming off of it, the functional groups, these variable side chains cause the amino acid to have a unique chemical property depending on what is coming off of it. So whatever the side chain does, it gives it unique properties and how it can function. It's like the alphabet, but instead of 26 letters, making all the words of the English language by rearranging them, you have 20 amino acids making all the proteins of the protein language by rearranging them. Let's look at these amino acids. You can find the same information on page 77 in your textbook. But here we have nonpolar amino acids. We have 10 of them. You have glycine, you have alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. I am not going to make you memorize the 20 different side groups of the amino acids. You will have college professors that make you memorize these side groups. It's fine. What makes these side groups nonpolar and hydrophobic? Take a minute to look at them. You have, of course, your amine group, your central carbon with a hydrogen coming off of it, and your carboxylic acid. So we already know that's not, none of that is what's making it nonpolar. So what is making it nonpolar? If you are saying it's because all of these side groups are mostly hydrocarbons, you are correct. We have methyl groups, those CH3 groups, hanging off of all of these down here with other CH2 groups in the way. And then here's nothing but carbons and hydrogens, Sure, we have an amine group here, and sure, we have a sulfhydryl group here, but overall, they're hydrophobic and nonpolar. That was 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. One of them does take the place of the other one, unless I just count it wrong. But then we have our polar groups, that these are charged polar, which means they are all hydrophilic. Polar, serine, threonine, cysteine, tyrosine, asparagine, glutamine, and then our electrically charged acidic and basic groups, aspartic acid, glutamic acid, lysine, arginine, and histidine. Why are these polar and hydrophilic? Take a second to look. If you said it's because it has these hydroxyl groups coming off here, the sulfhydryl group, the amine group, that gives them all polar, partial negative or partial positive charges, then you are correct. Same for down here, except let's talk about these acids and bases. Acids and bases work on ionizing cellular waters, so they help with the pH of waters. You have acids, amino acids with acidic side chains that are hydrogen donors, so they will decrease the pH of their surrounding solution. This is aspartate, glutamate, asparagine, and glutamine. You have amino acids with basic side chains, so they will actually take up hydrogen from their surrounding solutions and make it more basic, because as you remove hydrogen, you keep hydroxyl groups in the solution, and this is lysine, arginine, and histidine. Then you have sulfur-containing amino acids. We have 2 of them. We have cysteine and methionine. Sulfur, I told you back in the carbon lecture with the functional groups, forms one of the strongest covalent bonds that exist on this planet with itself. It's called a disulfide bridge. And we find it in proteins, and we find it in nucleic acids as well. This covalent cross-link between sulfhydryls stabilizes the 3D structure. Sulfur. Can you think of things that smell like sulfur? Have any of you ever walked into a beauty shop or a barber shop? Well, not barber shop so much, but a beauty shop, and it just smells like sulfur? That's probably something that happened back more when I was a child, not so much you, because it was something called perms. Perms were all the rage in the 80s and early 90s. Now, my hair is naturally frizzy, actually, but I currently have it straightened because I was bored one day. But hair. When you straighten hair, you chemically alter it by breaking the bonds of the hair. But you can change the chemical structure of hair by putting bonds back into it. That's what perms are. So it's a chemical alteration to the hair by adding bonds. And the bonds they add are disulfide bonds. And so they put, obviously this is a giant representation, but they'll put a supergroup in, and then that causes the hair to bind to itself, right? Well, okay, if it would hold. It causes the hair to bind to itself. I just have too much, that's the problem. Ow, ow, ow. Miss Segovia, if we could walk into each other's rooms, she has plastic models of this that works a lot better. But hey, I have actual hair. Well, so does she. She doesn't not have hair. Anyway, so it causes the hair to bind to itself and makes a bond like that. Obviously, once again, this is a giant representation of what's happening chemically in the hair. And then it does it again farther down, and it causes a bond that didn't exist originally to form a disulfide bond and bind to the hair. And your hair gets really kinky and curly. And that is how a perm works. The reason perms smell like rotten eggs and sulfur is because they're using sulfur to form these bonds. In the human body, these sulfur bonds are some of the strongest bonds that exist. Share screen. Yes, thank you. And we use that to stabilize the 3D structure. Perms smell like rotten eggs. Okay, in order to build a protein, you need not glycosidic linkages, not ester linkages, but peptide bonds. So here's the third bond in our macromolecules that we form through dehydration synthesis. Covalent bond between the amine group of one amino acid and the carboxylic group of another. So look here, it obviously involves enzymes. Oh, it's not gonna make me, okay. You lose the hydroxide of the carboxylic acid and the hydrogen of the next amine group as water. Boom, it's out, you dehydrated it, and you form a peptide bond. This occurs in ribosomes in the human cell. The ribosomes can either be free in the cytosol, which is the aqueous solution of the cell, or they can be bound to rough endoplasmic reticulum, which you'll learn about after your first exam. You can only build in one direction, carboxylic group to amine, carboxylic group to amine. You can't go the other way. It's an NHN and a carboxylic end. So it's NCC, NCC, NCC. I don't know if y'all are Star Trek fans, but the Star Trek ships all have NCC. Yes, no? See? Ha! I'm not crazy. I mean, I'm crazy. But they can only grow in one direction. Not the band. They can grow in many directions, I'm sure. But the amino acids can only bind to each other in one direction. Now, I told you that proteins have 4 different layers of structures: primary, secondary, tertiary, and quaternary. The function depends on that final 3D structure that occurs in the tertiary stage, and then if it needs to be bound to other polypeptides in the quaternary stage, that final 3D structure. It twists, it folds, and it coils into its unique shape. The primary structure is easy. It's just the amino acids in a chain. Every single polypeptide in our human body starts with methionine because methionine is the signal for the ribosomal unit to bind. So everything starts with methionine, and then everything that comes after it is just a rearrangement of the 20 amino acids to make our proteins. So it's the primary structure is the amino acid sequence determined by the gene. If you were to take your DNA and then of course turn it into RNA and then have that turned into a protein, and you didn't do any kind of folding, you would just have this one linear line of amino acids for however long it needs to be a protein. And that's the primary structure. A slight change in the amino acid sequence, so a mutation, can affect protein structure in its function. It can actually make it to where it doesn't function at all. There's quite a few genetic disorders that are caused by mutations of certain proteins. Even just one amino acid can make all the difference. If you put a polar amino acid where non-polar is supposed to go, then you have drastically changed the function of that protein. Lysozyme is an enzyme in tears and mucus that kills bacteria, and that's what you're looking at here. This is the protein for lysozyme. Once you have that single linear chain of amino acids, okay, so even just one amino acid can make all the difference, such as in sickle cell anemia. Sickle cell anemia is valine, histidine, leucine, threonine, proline, glutamine, glutamine. That's a normal red blood cell. In sickle cell anemia, instead of that number 6 glutamine, a valine has been mutatedly, accidentally placed there. That's a DNA mutation. The DNA is coding wrong, and it puts valine where glutamine should be. Instead of getting the nice donut-shaped structure of an adult functional red blood cell, you get this sickle shape. If y'all are not into any kind of mythology, the sickle is what death carries around, or Thanatos, if you like Greek mythology. He's always shown carrying a sickle. Sickles are farm implements that are used to harvest wheat. But here is the sickle shape. This sickle cell anemia is very fascinating in terms of the genetic mutation, because we believe sickle cell anemia, the genetic mutation derived in African populations as a way to prevent them from getting malaria, because malaria cannot infect a sickle-shaped blood cell. It can infect all these healthy red blood cells, but it can't infect the sickle shape. So it's a trade-off, because sickle cell anemia causes its own problems, like the cells getting all clotted up in the spleen or in the blood vessels, and you have less oxygen carrying capacity, but you don't die from malaria. So traditionally speaking, sickle cell anemia affects African American and African populations, because we believe that the reason it was created in the first place, the evolutionary reason, was to prevent against malaria. Right, so I told you, if you put a I did it backwards, but if you put a hydrophobic where a hydrophilic is supposed to go, you have drastically changed that protein. We were just lucky, evolutionarily speaking, that this didn't cause immediate death. It actually created something that's slightly functional, and so the person can still live. Secondary structure is the primary chain folding on itself. It can do two things. They're called alpha sheets. No, they're called alpha helices and beta pleated sheets. So it's folding along short sections of the polypeptide, and its interactions between the adjacent amino acids. And you see both alpha helices and beta pleated sheets in all secondary structures of polyproteins. It just depends on where they happen and how that protein is structured to function. So beta pleated sheets, of course, are like if you take an accordion and stretch it out, it's the wavy part of the accordion. Alpha helices, or an alpha helix. It's created by hydrogen bonding, which are, weak bonds between R groups, and form sections of a 3D structure. So here's an alpha helice. Here's a beta pleated sheet. Just remember over and over and over again. You have electrostatic attraction due to hydrogen bonding. And as we know, that's individually weak, but collectively strong. Tertiary structure is when all those alpha helices and beta pleated sheets start folding on itself. The whole molecule starts to fold into a giant structure. It's interactions between different amino acids. They can be hydrophobic reactions, so they push each other away. They can be, or hydrophobic because they have to be interior, so they stay inside. So they push the outside away, I guess is what I mean, because they have to be interior. You can have hydrogen bonds and ionic bonds and disulfide bridges. So overall, the tertiary structure of protein folding is actually fairly strong. That's actually it for each individual polypeptide. And then, oh, it anchors the 3D shape. And then you have the quaternary structure, which is more than one polypeptide bonded together. So if something needs to be hydrophobic, it has to go to the inside. Hydrophilic stays to the outside. So here's collagen in your skin and your tendons, and here's hemoglobin in your blood. So primary, it's just the chain of amino acids. Secondary is alpha helices and beta pleated sheets. Tertiary is the whole thing, all those alpha helices and beta pleated sheets folding on each other. And then the quaternary structure is multiple proteins or polypeptides working together. It says here, it says here, only when all these polypeptides working together do they become a functional protein. That is mostly true. There are a few proteins in the human body that function at the tertiary level. Not everything needs to be quaternary. So that's not a 100% rule right there. So the amino acid sequence is formed by peptide bonds through dehydration synthesis, and this is determined by the DNA. The DNA tells the ribosome what to make. Secondary structure is R groups bonding by hydrogen bonds. The tertiary structure is those R groups having hydrophobic interactions to the inside, so all this inside part would be hydrophobic. Disulfide bridges and hydrogen and ionic bonds, which are stabilizing the entire 3D structure. And then quaternary structure is multiple polypeptides. Once again, you have those hydrophobic interactions in the middle. Everything that's hydrophobic has to be to the middle. You can unfold a protein. You don't usually want to unfold a protein because it destroys the ability for it to function. But you can unfold a protein through conditions that disrupt hydrogen bonds, ionic bonds, or disulfide bridges. So temperature. You ever cook an egg? When you drop an egg in the pan, you have the yellow yolk, but then you have that clear albumin. And what happens once that clear albumin hits heat? It turns white, exactly. Well, that's because the proteins in that albumin are denaturing, because the temperature is causing them to unravel because it's breaking all the bonds. That's why the color change occurs. pH can also break the bonds, and too much or too little salt. It alters that secondary and tertiary structure, so therefore it alters the entire 3D shape and destroys the function. You don't want your proteins to denature. In biology, size doesn't matter, although it does, because everything has to be microscopic in our bodies, but shape is very important. So, you hungry yet? Let's go eat some proteins so that we can build proteins in our body. Thank you.